Hello everyone, a very good evening and welcome back again on the Proteomics Bootcamp IIT Bombay live webcast series. Today is the third session of this webcast, and today we have a very distinguished speaker, Dr. Sandeepan Ray. Sandeepan Ray has finished his doctoral work from IIT Bombay in Proteomics lab, and then he moved on to University of Cambridge for his postdoctoral studies. He's currently working in systems pharmacology and therapeutics at the School of Medicine, University of Pennsylvania. He's also visiting scientist in the Francis Crick Institute. He's currently working in circadian rhyme and trying to apply the multiple omic technologies to understand the physiological problems. Today's topic will be the multiplexed quantitative proteomics for mechanistic study of circadian rhymes. So Sandeepan is going to give you a real flavor of how to apply the proteomic technologies to address the biological questions of interest. In the process, he will also explain you the experiments, what he has performed for doing quantitative proteomics, and then how this kind of data set could be utilized for obtaining the meaningful biological insights. So please welcome Dr. Sandeepan Ray. He's currently in USA, where working in a different time zone, but he's still ready to chat with us and share his work. So I'm going to share our screen, connecting him on the Skype directly, and he's going to give you talk. Additionally, if you have questions, we will take at the end. Please do share the question on the portal.
So now I'm starting the screen share.
Yes.
So are you able to see my slides?
Yes. So it's now, you can take over and start presentation.
Okay, so very good evening to everyone. So it's a great pleasure to talk about our research work that we are carrying out at this stage at University of Pennsylvania. And I am very thankful to Professor Srivastav for organizing such an interesting event where we can share our work. It's indeed a very good platform where we can share our work, we can have queries, we can share new technologies, as well as we can get feedback from others on our research. So I think it is meaningful and very much appreciable. So today I will talk about multiplex quantitative proteomics for mechanistic study of circadian rhythms. So are you able to see my second slide? Just to
Please continue.
Okay, so thank you. So what are the circadian clocks and what is their implication in health and disease? So we reside on a planet that have an intrinsic rotation period of nearly 24 hours that in turn embedded nearly 24-hour rhythm in all living organisms, whether it is starting from very primitive archaebacteria to the higher mammals. And so it is an evolutionary conserved trait that is controlling the physiological and behavioral scenario in all the living organism, including very primitive archaebacteria to the higher mammals like us. So perhaps you have noticed that our daily physiological processes are work in a rhythmic pattern, and even before we wake up in the morning, it makes our body suitable for the day, and the release of the cortisol, increase in the blood pressure and body temperature, and that's make you understanding that the day is starting, and with the starting of the daylight, your high alertness and best coordination during the noon time, then what is the best time to work, and then again at the end of the day when it is dusk time, your body temperature slightly going down, your blood pressure starting going down, and the melatonin secretion is high, whether your body is now going to a sleep state, and that's going to a deep sleep in the midnight. So it happens in a cyclic manner, and which is known as circadian rhythms or biological clocks. And the circadian aberrations, where your normal clock is disrupted or aberrated, that increase the risk of several diseases, including cardiovascular disease, risk of diabetes, different type of cancers and metabolic disorders, sleep disorders. So that clearly indicates that how important for us to maintain a decent body clock. So without going to the details of these studies, if we see some of the news headlines in BBC or the Telegraph, so it's showing that how the circadian rhythm is interpreted and implemented in the daily basis, or whether it is infection by an external agent, or it is the healing of the wounds, or even it is the timing of the surgery. So this is all are the studies that published in some reputed journals and showing the implications of the body clock, and it's clearly indicating that we are not equally active or equally susceptible or equally immune to the environment or the infecting agent throughout the day and night. And it makes sense to understand that what is driving this force that maintaining a daily rhythm in our physiological and behavioral processes. So the Nobel Prize in Physiology and Medicine in 2017 clearly indicates the immense implications of this field in clinics. The three pioneer scientists of our field, Professors Jeffrey Hall, Michael Rochbass, and Michael Young, was given this Nobel Prize in 2017 for their intellectual work in the mechanism of biological clocks and how that is connected to human health and diseases. So our lab at the University of Pennsylvania is interested in biological clocks and sleeps and their interactions. We do work on molecular mechanism of non-transcriptional circadian clocks, redox oscillations in circadian clock, mechanism of action of some drugs that can modulate circadian rhythms, and also the crosstalks among these physiological processes using systems level approaches. So today I will focus on the molecular mechanism of non-transcriptional or circadian clocks, and also the mechanism of some drugs that can modulate circadian rhythms for therapeutic benefits. So part A of my talk will talk about some non-transcriptional, non-canonical circadian clocks, which is not necessarily follow the regular mechanism that is proposed to explain the circadian rhythms. So in general, the circadian rhythms is considered to be governed by transcriptional and translational feedback loops, where there is a positive and negative regulators. The positive feedback loop promotes the transcriptions of the negative elements, and the negative elements in turn inhibits their own transcription and translation that make a loop stop. And again, when their levels are going down, then again the positive regulator comes in play and the clock starts again. And the components of the clocks are different in different organisms, whether it's in a Drosophila, it's par, team, clock and cycle, and while it is the higher mammals, we have par, fry, clock, bimal 1, etc. But while the world believes in the circadian rhythms are completely driven by transcription translational feedback loop, in our laboratory a couple of years ago, we first proposed that the circadian rhythms even can exist without a transcription translational feedback loop, and that is happening in our red blood cells. where the transcriptional translational feedback loop is absent, but still we identified the rhythmic pattern in the oxidation cycle of peroxyredoxin proteins. And this type of work happened in 2011 and 12 before my joining in the lab. So these were two subsequent Nature papers on the findings that circadian rhythms can exist without transcription in eukaryote, that is in the human red blood cells. Subsequently, we have also shown the oxidation cycles of peroxyredoxin proteins are conserved across the phyla. That is, it is conserved from very primitive archaebacteria to higher mammals, indicating that, so this is proteins are conserved across the phyla. Hello?
Yeah.
So, is it going on fine?
Yeah,
I am completely audible?
Yeah, complete.
Please continue.
Thank you. So this is conserved across the different phyla, starting from archaebacteria to higher mammals, and indicating that peroxyredoxin proteins could be a marker for circadian rhythms, which is evolutionary conserved. And this was another Nature paper that indicating peroxyredoxin are conserved markers of circadian rhythm. Okay, so now onwards we concentrated on the omics level approaches to study the circadian rhythms at a omics level, where we wanted to see what is happening in the transcriptome, proteome, or metabolome level in the mutants, clock mutants, which is generally considered to be don't have a behavioral rhythm. To test that, in around 4-5 years before, we started work in a Drosophila cells that lacking the clock gene. This is Snyder cell lines, so which is believed to behaviorally arrhythmic, but we wanted to see that whether these cells still have any rhythmic pattern in their transcriptome, proteome, and metabolome level. And it was an article that we published last year in Molecular Systems Biology. In fact, it was the cover page article of the journal, where we show that the rhythmic transcript, rhythmic proteins, and rhythmic metabolites are present in Drosophila Snyder cells that lacks a core clock component. Now I will discuss in more details what is our ongoing work and two levels of applications of proteomics technologies, so that helping us to decipher the complex biological processes like circadian rhythms, and also at the end of my talk I will discuss how some of the drugs that can be used to manipulate circadian rhythms and could be therapeutically beneficial. So with these results, we propose that while the TTFL or transcription translational feedback loops are the key mechanism in driving transcriptional oscillation, there are post-transcriptional and post-translational oscillator as well, which we call the NTOs, which is non-transcriptional oscillations. And there is an interaction between two models, and there are some more work to do to find these NTOs and identify the mechanism behind this that drives these non-transcriptional oscillations. So now if I go back to my slide number 2, where I shown that the circadian clocks work to a positive regulator and negative regulator feedback loop, and concentrate in mammal that the components of the positive and negative feedback loops are clock and BMAL are the positive feedback loop that driving the transcription of period and cytochrome that in turns inhibits their own expressions and making the second negative feedback loop. So now we are considering whether a system that lacking a core clock component like BMAL1, whether those have still any rhythmic pattern at the proteome, genome, or phosphoproteome level. So just to give a brief description of the protein BMAL1, or which is known as ARNTL in mouse. So it is a transcription factor that we have seen the principal driver of the circadian clocks in mammalian system. And a recent study has also shown that it is a moonlight protein that can also act as a translational factor and regulate circadian rhythm at the protein synthesis level. And what is observed from multiple studies, the targeted disruption and deletion of BMAL1 caused the abolition of behavioral rhythm, aberration in sleep-wake cycle, neurodegeneration, and reduced activity and lifetime. However, it is not clear that whether this protein is absolutely essential for driving the rhythm at a genome scale. To answer that, we first need to understand the neurological machinery in the mammal, where the suprachiasmatic nucleus controls the rhythm all over the body. So even there is a rhythm absence present in the peripheral tissues, there is a high possibility that the signals from the brain is suppressing that, and the molecular rhythmicity is not visible at a behavioral level. To check this, we started our study with two types of cell line, MSF, that is mouse skin fibroblast cell line, and organotypic culture liver tissue samples from mice which are wild type, that is having both the copies of the BMAL as active level and also the double knockout mice, which is BMAL double negative, which is thought to be behaviorally arrhythmic. But we want to test that whether still there is any rhythmic pattern in their genome level, which can propose some additional mechanism which is not presently known. So to do that, we've taken the advantage of high throughput technologies like whole RNA sequencing, quantitative proteomics, and quantitative phosphoproteomics, and then we analyze the rhythmic pattern of the molecules at transcript level, protein level, and phosphoproteome level, and we identify the rhythmic pattern indeed present even in the absence of this core clock component. So now I would like to move more details of these studies and show some practical applications where proteomics or quantitative proteomics enables us to get the intricate details of complex biological processes like circadian rhythms. So first we checked the expression levels of the core clock components in the wild type and knockout system, and as expected that it is showing a rhythm in the wild type, while it is the rhythm for the core clock component is completely lost in the knockout. But when we are doing a complete RNA sequencing, surprisingly we observe that the RNA level expressions are rhythmic even in the absence of the core clock components, and more interestingly, those rhythmic components are almost mutually exclusive in wild type and knockout. That indicating that there is something driving force apart from the core clock components, which is considered as the prime driver of the circadian rhythm. And interestingly, with a very high false discovery rate, that is very stringent false discovery rate of 5%, we're still able to identify thousands of rhythmic transcript in the BMAL knockout systems, which is in general thought to have completely arrhythmic in terms of a behavioral pattern. Now we tried to drive the molecular mechanism that is driving the transcription level oscillations in the absence of the core clock, and we are able to identify few motifs that could be driving the morning and night phase transcription oscillations, and those are mostly restricted to the ETS transcription factors, which is a very novel discovery because the ETS transcription factors were not known to be a driving force as the circadian rhythms. And interestingly, we found the PRX protein, that is the peroxiredoxin protein, that is also oscillating in a BMAL double negative system, indicating that there is a parallel procedure that is happening in the systems that's lacking the core clock components, but still molecular rhythms are existing at the transcriptome level. So now we try to see whether we can also see the rhythmic expression at the proteome level. To do this, we tried with the multiplexing technology where we use the tandem mustags. Well, since I am talking to a proteomics community, I anticipate many of us are familiar with these technologies. So this is a multiplexing technology. There could be 6-plex, it could be 2-plex, it could be 10-plex, depending on the need. The beauty of the technique is that you are labeling the proteins from different time point, or whatever it is your test and control population. It could be drug treated and non-treated, and you are labeling those with different TMT tags, and you are pulling that and running in a same mass spectrometric run. What are the advantages? Advantages are manifold. Number one, it saves time and mass spec time and missing values, because since you are running the all 10 samples in the same mass spec run, there are reduced missing values, better statistics, and most importantly, there is less run-to-run variation, which we often observe while we are running the samples separately. So particularly in this type of experiment where we are analyzing 20 time points across 3 days time, it needs extremely robust technology to identify the molecular oscillations where the differences are often very subtle, and you need to identify that even a small level of ups and downs in the protein expression level, and that this multiplex quantitative proteomics allowed us to do so. And just to give little brief of the techniques, that the TMT reagents, that's basically composed of a mass reporter, mass enhancer, normalizer, and NH2 reactive group, so that binds to the lysine epsilon residues, and with the balancer group, it's actually given a 1 tone difference that could be detected in the MS2 spectra, and you can quantify your different conditions labeled with 10 different TMT levels. So now moving to the actual results, as I mentioned that this multiplexing allowed us to get reduced variations and great reproducibility even with the biological replicates like the circadian replicates, that is the protein expressions at the time point 0 versus 24 or 48 hours. We got a very good correlations in terms of protein expressions and very few missing values that allowed us a better statistics. So as we have observed, hello,
yeah,
is everything going okay?
yes, sir. Continue, sir.
Okay, thank you. So as we observe a rhythmic pattern at the transcription level, so we observe the rhythmic pattern at the proteome level as well. And if you see the curves that across 54 hours, that is more than 2 days, we are getting the protein pattern in both wild type and knockout systems, which is definitely out of the box result, because it is considered that the core component, when it is deleted or depleted, there is no rhythmic pattern. But we challenge this paradigm and we are telling that okay, it might be the core clock components are not rhythmic, but still there are rhythmic pattern, and when we are doing it at a global scale, we are able to get meaningful components of the body which is actually showing rhythmicity even absence of the core clock machinery. And interestingly, when we compare the cellular component, molecular functions, and biological processes in the protein and transcript level, both in the wild type and knockout, we observe very high levels of overlaps among these geo terms in the protein and transcripts. That indicates that the rhythmic proteins and rhythmic transcripts are actually in the same directions and indicating a good level of validations of these two different findings at different levels. So interestingly, when we trying to find out what is the biological processes associated, we identified the mitochondrial cellular component, and the biological processes involved in metabolism is highly enriched both in the liver and skin fibroblast in the knockout system compared to the wild type, and both in the protein and transcript level. So indeed it is showing the need for the rhythmic metabolism in the absence of the core clock components, and which might be driven by some components of the mitochondria that is not presently known to drive such oscillations. Further, in the same flow, we try to see whether there are still rhythmic pattern at the post-translational level, and we try to cover the phosphoproteome profile in the bimal double negative systems using the similar technology where we divided the stuffs in the discovery and validation stage, and we measured the phosphoproteome in 3-hour resolution time across 2 days, and to see that whether there is any rhythmic pattern in the phosphoproteome level. And using the TiO2-based enrichment method, we were able to detect nearly 6,500 phosphosites and measured their abundance across 48 hours. And interestingly, again, we find a rhythmic pattern in phosphorylation in the bimal double knockout system. And interestingly, as I mentioned before, these are many new phosphosites which are coming as rhythmic in the knockout system. Those are not rhythmic in wild type and vice versa. That's indicating that there are still the physiological processes are running in rhythmic fashion, which may not be detected when we are detecting some core clock component, but in an unbiased screening, when we are screening the whole proteome, whole transcriptome, or whole phosphoproteome, we are able to detect these rhythmic patterns. And as we have seen in the proteome, we can see the rhythmic pattern of phosphorylation across the 48 hours time, and it is showing a very good rhythm in the discovery set and the validation set of 3-hour resolution time point collection. So now we question that if the bimal-1 is still bimal-1 knockout systems are still showing a rhythmic pattern in the proteome and transcriptome level. So what are the reason for having a behaviorally arrhythmic animal when the bimal is knocked out? So to take that, we did the quantitative proteomics and phosphoproteomics study of the in vivo in 3 wild type and 3 knockout mice, and identified that the bimal knockout affects the hepatic proteome in a drastic manner. Nearly 25% of the identified proteins were altered. So it's surprising that many of the metabolism key core metabolism techniques as processes, as well as the translation and energy generating processes, are affected due to this knockout. So with this, we also identified that there is a lower level of phosphorylation when we do the total phosphoproteome patterning on a gel, that is scanned with ProQ Diamond, and then the total protein with SuproRuby. The total phosphoproteome level is lower in a bimal knockout system, which was further validated by the mass spectrometric analysis, and we identified that nearly 500 phosphosites are altered due to this knockout. And again, those are involved in very key processes, and indeed the bimal is an interactor of many known kinases and phosphatases and their regulators. So it makes sense that this knockout of the bimal-1 protein can affect the physiology in a very severe way than what is actually believed at present, that the effects are completely due to its clock activity. But we propose that it's not the clock activity, it's far beyond the clock activity, and it's actually affecting the whole kinome profile of the organism, and that actually giving the irregular behavioral pattern that we observe in the animal. So to check that, we used a very recently developed method, the destabiotin nucleotide probe-based enrichment of ATPase enzymes. That is actually a semi-selective method for kinase enrichment, where in the knockout and the wild type, we use these probes to enrich the ATPase enzyme. So these probes of 2 types, that is the destabiotin ADP and ATP probes, that selectively binds to the active residue of the lysines and allow the selective enrichment of those. And then in the mass spec run, you can selectively see the metabolically active enzymes and kinases, and many of the low abundant kinases which are not measured, could be measured in a normal proteomics analysis, could be measured. And this allowed us to quantify nearly 200 kinases in the mammalian system, and many of which actually shown were differentially altered in the knockout. And interestingly, when we compared the kinases identified our study with the core, with the binding site of this core clock component, which is a science paper decades ago, we find a very good overlap with the chipset data with our phosphoproteomics data. So we categorize the kinases in 4 levels: those which are identified in the global proteomics level, those which are identified to be altered in the one described with enrichment level, and also the phosphoproteome shown the expression levels of many phosphosites are altered. We did a kinase substrate database analysis to identify the kinases that are associated with those phosphosites and made a comprehensive kinome map in the Bimal knockout system, which indicating that several kinases involved in ERK and MAP kinase signaling, Hedgehog and Chor and RAF signaling are severely affected by this knockout, and that can cumulatively make a phenotype that is behaviorally arrhythmic or having a lesser lifespan or the activity. So with this, we propose that the Bimal1, which is considered as the prime driver of clock system, is basically not required for self-sustained 24-hour oscillation, and molecular rhythms are plastic that can reconfigure themselves to compensate the lack of the core clock components, and also with our analysis the ETS transcription factors and redox oscillator like PRDX protein came forward that may be the drivers of the molecular rhythms in Bimal double knockout systems. And also we propose that Bimal1 regulates diverse physiological processes through kinome networks, and that may attribute the physiological abnormality shown in this Bimal knockout animals. And we propose the mechanism when the core clock is absent. It might be the non-transcriptional redox and the transcriptional ETS oscillations are coupling and driving the oscillations at a genome level. Now I will move to the second part of my talk, where we can see some more application of proteomic technologies for deciphering the mechanism of actions of some interesting molecules that can help to alter the circadian period length and enable us to treat the circadian misalignments and disruptions. So from the first part of our talk, we have seen that circadian rhythms is immensely important for sound health, and it has implication in different diseases. So subsequently, so the small molecules that are capable of modulating circadian rhythm must have some therapeutic implications. So there was a couple of studies in 2008 and 9 where the library of pharmacologically active components were screened, and some potential components were shown that can modulate circadian rhythms. So we selected 9 potential modulating drugs from those and tested those on human osteosarcoma cell line, which is a very well-known model for circadian rhythm. So we categorized the drugs in 2 groups. One that can enhance the period length, that is like longdacin, roscovitin, purvacanol A or SP600 125, and in reciprocal there are drugs that can shorten the circadian rhythms, and there are many candidates in that, and we tested how those drugs exactly work, which is presently not known. So we tested the effect of drugs on the circadian period length in U2S cell line, and we find that the drugs can change the circadian period in a dose-dependent manner, that implicating that when there is a circadian aberration in a patient having a sleep disorder, or it's due to jet lag, where the circadian or for the shift worker who often have the circadian aberration due to their change in the day-night cycle due to professional reason, could be treated with these drugs that can that can allow to modulate the circadian rhythms in a dose-dependent manner and can correct the aberrations that may happen in the circadian daily clock due to different physiological processes that are showing abnormality, or like due to the shift work or sleep-related disorders. So to check that how these drugs exactly work, again we did a quantitative proteomics using the 10-plex quantitative proteomics method using the tandem mass stack, where we compared the DMSO vesicle as a control, and the four drugs, Longdacin, Purvacanol A, Rofcovitin, and SP600-125, we tested the effect on U2S proteome. And interestingly, we find that if we see the pie chart, many of the altered proteins are signaling molecules or kinases. Further, if we see the proteome maps where we are plotted the the associations of this altered protein in biological processes, we can see the ribosome that is associated with translation, and as well as different kinase pathways, whether MAP kinase pathway and other kinase signaling pathways are commonly altered by these drugs. So further, we did the in-depth phosphoproteomics analysis and find the alteration of different phosphosites, which are mostly lower level of phosphorylations in the presence of these drugs. And if we see that there are many of these altered phosphosites are common among these four drugs, and particularly if we see some of the phosphoproteins which are multiple phosphosites, which are showing the, if we see the lower level panel of this slide, so those are actually overlapping targets for these drugs and showing a reduced level of phosphorylation. So when there is alteration in phosphorylation, there is a meaningful to study the kinases which are actually altered by these drugs. And indeed, if the kinome profiling, as we have shown my previous slides in the previous part of the talk, are very good complementary analysis of the phosphoproteome, where we can say, okay, this phosphoprotein is altered and whether the corresponding kinase activity is also altered. Using the same destabiotin nucleotide probes, we profile the kinome in presence and absence of the drugs, and these drugs that binds to the kinase selectively compete with the probes, and when there is a drug binding, in the mass spectrometry we will see a lower signal for that particular kinases. And with this method, we are able to quantify nearly 200 kinases across the four drugs and compare their fold change against the control. And interestingly, some of the kinases, as we will see in the subsequent slides, are commonly inhibited by these drugs. And we identified the nearly 8 kinases which are commonly inhibited by these drugs, and most of them are lying in the CAMK, which is the calcium and calmodulin dependent kinases, and CMGC group of kinases that comprise the CDK, the casein kinase group, and also the GSKs. So it's indicating that many of the kinases are commonly inhibited by these drugs, and if we map those in the kinome map, we are able to find the associations among these kinases. So now, since it's a new technology, we need to validate our results using a completely different method. So we tried to do that by hotspot radiometric assay. So it is a method that developed in 2002 by Reaction Biology Corporation in collaboration with some academia. This is a comprehensive assay for kinase catalytic activity using some radioactive substrate and ATP, where you can selectively measure the kinase activity 1-to-1 with the substrates. And we use this method to substrate to using substrate and radio level ATP and measure the direct activity of some interesting kinases that identified in our mass spectrometric quantitative proteomics. And interestingly, we found very good correlation with our mass spectrometry based measurements, and indeed many of these kinases are equally inhibited by these drugs and targets like CDK7, casein kinase 1, casein kinase 2, MST4, JNK. Those could be a common targets for these drugs, and subsequently we are able to do a 10-dose curve from where we can able to detect the IC50 of these drugs for these particular kinases and compare those to a known kinase inhibitor, staurosporin, and we found that there are overlapping kinase targets for this drug which was not previously known. And more interestingly, when we searched the circadian period length with the knockdown of this particular target kinases using siRNA knockdown method from the BioGPS circadian database, interestingly, all these knockdowns leads to a longer period lengths that completely validate that our identified targets make sense in terms of their subsequent effect in the period length. And interestingly, when we plotted the altered phosphosides and the kinases, and along with the core clock components, we were able to detect a very good interactions among those, and which leads to the alteration of several signaling pathways, starting from BCR signaling, MAP kinase, HOG, and AMPK signaling. And in IPA, we are able to detect a very good interaction among those signaling pathways, and that's actually altered by these drugs. And if you see the yellow dots inside the particular altered phosphoproteins or kinases, those indicators that the siRNA based knockdown of those proteins leads to a longer period length. That is actually a very good validations of the proteomic based studies. So the last part of my study, I will just show a very new technology that's actually came forward a couple of years ago to tracking the potential molecular targets using a quantitative proteomics method. So as we know, when the drug binding to a protein stabilizes its thermal stability, so basically the TM value of that particular protein is increased with the binding of the drug, and which is called the CHESTA assay, that is the cellular thermal stability assay. So using the same principle, Mitchell Savitsky and colleagues developed a quantitative proteomics method where the whole proteome is scanned in the presence of this drug in different 10 temperature points, versus starting from 37 to 67, and the thermal curve of each protein is analyzed to check the direct binding of those proteins with this drugs. So particularly you can see that an unaffected thermal curve when there is no binding of the protein with the drug molecules, and that is for the 99% of the proteins you will see. Some will be showing a positive stabilization, that is a positive increase in the thermal stability, that is indicating those could be a potential direct binders of those drugs. And also there could be a second destabilization, that could be a secondary effect. We found very few compounds showing that because of the stability of one compound to another. And while selecting the direct binding candidates, we use very stringent statistical criteria that the proteins must be identified in 4 sets. We do it in 2 biological replicate tests and 2 controls in all 10 time points. So basically there are 40 different samples, and it need to be identified in all the set. Minimum slope should be less than minus 0.06, and melting curve difference in a control versus treatment experiment should be greater than the melting point difference in a 2 control systems, and the FDR corrected p-value must be 0.05 in at least one set of the experiment, and melting point shift should be the same sign, that is the positive, showing a stabilization of the protein. So we checked the thermal proteome profile of all these 3 drugs against the control using a thermal gradient from 37 to 67, and again we use a TMT based quantitative proteomics method to reduce the variations among the sample runnings. And interestingly, we are able to identify some known and some new candidates for this direct binding of these drugs. Like if we see the kinases like CDK6 or CDK1 or CK1D, those we identified in phosphoproteome or kinome profile. Since this clock regulatory kinases also showing direct binding with these drugs, and also we are able to identify a completely new set of binding partner like FBOXO21, RPL37, those are showing a positive shift for all the drugs, showing some overlapping candidates. And indeed, we are able to identify a handful of overlapping candidates which are showing very correlated thermal shift in all these 3 drugs. And more interestingly, those candidates, when their siRNA based knockdown assay, these are showing a longer period, indicating that we are able to track the correct hits using a quantitative proteomics method, which indeed is a very unbiased platform to track the drug targets across the whole proteome rather than a single substrate or binding assay, which is generally low throughput and cannot give an unbiased result like this. And with this, I will finally conclude. This is the fast proteome level investigation on circadian period modulating drug, and we identified differentially expressed protein as kinases, kinase regulators, ribosomal proteins, and proteins involved in translation and cell death, and altered phosphorylations of several of these identified proteins at the signaling pathways have we seen, and some of the proteins like CAMK1, CDK2 or 7, casein kinase 1, and MST, those are commonly altered targets for drugs. Those could be used as finding new drug molecules for the similar activities. So with this, I would like to end and acknowledge our group head, Professor Akhilesh Reddy, and my colleague, Dr. Uttam Valakunja. Most of the studies were performed when our lab was situated at the Francis Crick Institute. I would like to also acknowledge my colleagues at the mass spectrometry facility and some of the stuff we did with collaboration with University of Bristol, and last but not least the funding agencies including Wellcome Trust and University of Pennsylvania core funding, EMBO senior fellowship to our supervisor, and the Thermo Scientific TMT Mustang Award to me last year. And thank you for your patience, and I would be happy to take any questions on my presentation.
All right, Sadipan, thanks for a very nice presentation. And I think we can take some questions.
Yeah, so
first of all, I think it was, let me just start talking to you directly. Okay, so it was really interesting to see, like, how best you have made use of various quantitative proteomics technologies, and also try to understand the biological questions, using those methods. And especially something very, new, which you have tried out to look at the entire kinome using the just thiobiotin and nucleotide probes, right? So,
exactly.
So that how widely it is used, and how much you would assume for the various human sample. Have people started using this, for the kinome profiling?
Well, this kinome profile, there's kits that came in market a couple of years before, and probably you heard about John Rogers that we made in the last HUPO conference. So they have an application note that they couple this with the TMT-based quantitative proteomics. So the direct applications of these probes is still limited, because the thing is that it's not that widespread yet. But in our own research, and personally, I know many of the other research groups were using these technologies, and I am anticipating there would be a good numbers of papers coming with these techniques in coming years. So indeed, from a practical point of view, it's able to quantify many of the kinases which are not quantified in a regular proteomics workflow. And as you have seen that a completely different method, the radioactivity based assay that we did to validate the results, we obtained good correlations with the kinases that altered in the thiobiotin based method compared to the radioactivity based hotspot method. That is a very well established method for kinase activity. So we are getting good correlation. So we are thinking that this is a very good complementary method to analyze phosphoproteomics data and make a kinase substrate interaction sorts of stuff.
All right, that's great. Additionally, I think, thermal proteome profiling also you mentioned. So how widely it is used for the drug?
That is actually very widely. There is actually quite a few Nature and Science papers in last 3 years. So basically that was first proposed by Michael Sibisky in 2014. There was a science paper that targeting the drugs, and the beauty of the study is that you can do it in the living cell. Like it's not the cell extract. You are treating with the drug and it's in the living cell. So there are a couple of Nature protocol, Nature method, and Cell papers following that. So it's a very widely accepted technology now, and in the last 3 years there will be very high levels of high impact publications using this technique.
I'll just see if there are some questions on the portal. So looks like there are a couple of comments for you. One question is being asked that is it important to take equal number of discovery and validation samples as you have shown in the study? So I think some idea audience want to hear from you the quantitative proteomics experimental design, in which way you plan the TMT 10-plex workflows.
Well, the thing is that it's a very important question, because the validation phase and the discovery phase, which is often very important stuff. In the discovery phase, in a study like this, where we are doing the profile across the day, even at 3 samples, biological replicates is fine. But if at the same time we are doing a screening of a disease, for say, like what we did in IIT Bombay during my PhD, the screening of more candidates are required because the level of variations are much more. So I would say that a power calculations in the discovery phase study is very important, where it can show that at a 80% confidence level, the minimum number of replicates that is used in the discovery phase makes sense, and it is at least higher than the minimum number of candidates, which is in general for the 2D dice based method we found at least 8 candidates per group in case of a disease to control sample. And in terms of the validation phase, there are lots of opinions and stuff. Sometimes it's more desirable to do the validation in a different method, like it could be a SRM based method, it could be, and fortunately or unfortunately yet people wants to more see a completely different method like a immunoassay based method where we're doing Western blotting or ELISA or immunohistochemistry to validate the proteomics results, and the same time doing a radioactivity based assay that is completely different, is not dependent on mass spectrometry based assay. So that's actually give the confidence even higher. But at the same time, there are very good studies coming with the SRM based methods and stuff. So the validation phase and the discovery phase number of samples not necessarily need to be the same. In the validation phase, when you are doing a different immunoassay based technique, the validation could be in a big higher population, because that is much easier to do for say 100 of samples in ELISA plate. So it completely depends that what type of technology you are selecting for validating and what type of the validation, how good correlation you are getting with your discovery to validation phase, how much variation are there in your sample to samples. When there is a variance is high, you need to do it in a more sample. When your fold change is low, you need to do in a more samples to show that it is a stable difference.
All right, I think it's very much question the specific what biology question we want to address, and also the type of sample which we want to analyze. Accordingly, this will vary.
Exactly.
And that's very subjective. So thanks for the comment. Now, next question is from another participant. How can it be possible to study multiplex quantitative proteomics for mechanistic study for circadian rhythms? So, pretty much you have to summarize, your
Well,
work in some way.
the thing is that the main challenge in studying circadian rhythm is that it's not a test versus control. Like you are analyzing a disease versus control, or you are analyzing a comparison between drug treated versus treated, where the changes of protein or transcript level is much higher. But you need to understand that we are here analyzing the same subjects or same systems protein across the day. So the level of changes are comparatively much lower when we compare to a test versus control samples or so. So in that case, the quantitative proteomics is very important with a multiplexed method where the variations is much less and you are comparing the time frame across in the same mass spec run. So your deviations are much less. So getting something mechanistically useful, you first need to be very sure your technology is very robust, because the thing is need to be reproducible. It need to be making sense in terms of known knowledge, and particularly when you are proposing something new, you also need to show that some of the stuff you are identifying is actually matching with your known literature or known knowledge, and while you can add more information on the basis of that. So indeed, probably if you ask how it could be used in mechanistic study of circadian rhythm, it is the presentation I have given. That's you can see that the proteome level alterations across the day. You can detect the drugs that can alter the circadian rhythms, how those working, and also Matthias Mann's groups and Charlo, Maria Robles, and Mann, they are also doing lots of quantitative proteomics on circadian rhythms and sleep. So in 2017, they published the first phosphoproteomics level rhythmic pattern in cell metabolism. So these are other groups are also working, although it is much more limited compared to disease or drug. But there are studies that are coming that are very insightful in terms of giving the new as well as new, I mean, that's validating the earlier findings as well as giving very new openings that is actually not known previously. It's actually adding much more on the transcriptome level studies that happened in last 2 decades, while the proteome level studies are coming in place in last 8 to 10 years, I would say. Basically, the first hepatic proteome paper was published in 2006 by my research postdoc supervisor in current biology. That was a 2D diet-based method, but he was able to detect the peroxyredoxin oscillations that in turn lead to the three Nature papers in future when we identified that protein. But the initial identification was a proteomic study in 2006. So basically, even a high throughput screening can give a very good hit that could be studied further.
All right. Thank you so much, Sandeepan, for very insightful work and giving very detailed response to the participants' questions. Uh, so this was Dr. Sandeepan Ray from University of Pennsylvania, talking to us today about use of multiplex technologies to study biological questions in the areas of circadian rhymes. We will have this, webcast series every Thursday. As promised, we will have more industry presenters as well. So next week we have a industry presentation to talk about new technologies in the areas of proteomics and how your proteomics-based research could benefit from the advent of new methods and new technologies. Thank you very much for being with us,
Thank you
and
very
we'll
much.
join you again next week. Thank you.